Hello, everybody. Yes, oh my God, it's so wonderful to see you all milling about there. It tells me that we've done our jobs. We've given you a night of drinking and thinking. Now, of course, as we drink, we inevitably have to pee. And yeah, oh yeah, one man is brave enough to clap for peeing. Yeah, all right, oh, there we go. Yay! All right, so very excited. We got our final talk of the evening, and I have to say I'm very excited. All of these talks have been fantastic, and I'm really thrilled to close out the show with what is going to be a very cool topic, very near and dear to my heart as a man who loves microbiology. So we're going to hear from Dr. Krystal Thomas-White, a doctor! Has been studying the bladder and vaginal microbiomes in the context of women's health for over 10 years. She is now the senior scientist at the women's health startup Evvy. Evvy. As well as a dedicated science communicator and scientific advisor for Live UTI Free, a patient advocacy group. Please welcome to the stage, Dr. Krystal Thomas-White!
Thank you. Hello, everyone. I am so excited to be here because I've had this talk ready and waiting to go. I was supposed to give it March 2020, and we all know how that turned out. So I am finally here in front of you all to talk about pee, and I'm very excited. Quick about me, like you said, been studying this field for now 10 years, which I can't believe. First in grad school, then at Stanford, and now at Evvy. But the main thing you need to know about me is that I'm a microbiologist. It means I spend my days thinking about organisms smaller than you can see with the naked eye. But my real passion is women's health. And so I've been working on this interaction between women's health and the microbiome. So let's start there. What is the microbiome? And everyone's probably heard of the gut microbiome. It's kind of having a moment right now. But the microbiome refers to all the bacteria that live in and on you. We're finding they have a huge effect on our health. And in about 2010, there was a national effort to try and map all the different areas of the human body, from the mouth, the gut, the skin, the genitals of both sexes. But one place they completely skipped was the bladder. I know. Why? Because, of course, urine is sterile, right? No. Who has heard that before? Yes, exactly. We all have. And I can guarantee you that every medical professional has heard that at some point in their training. So when I was in grad school, I was like, well, where did this idea even come from? How did it start? So I searched the literature and went all the way back to the grandfather of microbiology himself, Louis Pasteur. So Pasteur did some beautiful, elegant experiments that actually proved bacteria existed. And in those experiments, he used urine. And he found that a sealed jar of urine did not turn cloudy when it was left alone, meaning that no bacteria grew. And that led to this wonderful quote in an 1881 paper that fresh and healthy urine is perfectly free of bacteria or other minute organisms. I love this quote because it could have been written today, and it was written over 100 years ago. And before I go any further, I want to explain a little bit about microbiology and how we measure bacteria because they are so small. We like to grow them on agar plates. And what you do is you take a very little amount of your sample, you spread it out on the plate far enough so that one single cell will turn into a single colony. It's called a colony-forming unit. And in this way, we can measure the exact amount within a given sample. And if we take a look at our history from 1881 and we jump all the way to 1950, there was a doctor called Edward Cass that tried to use this idea to identify who was at risk of dying of a kidney infection. So when you have a bladder infection, it's possible for the bacteria to move into the kidneys, cross over into the bloodstream, cause sepsis and death. And he wanted to figure out who needed to be treated before they had surgery. So what he did is he took 1 microliter of urine, put it onto this type of plate, and said, well, if you have about 10 to the 5th CFU per mil, that patient should be treated before surgery, otherwise they may not survive. Now, this test was amazing. It saved lives. It was a huge success. And because it was so good, it got applied to absolutely everything, even things it wasn't designed to test for. So over time, the message got distorted. Physicians saw reports of no growth for people without symptoms and growth for people who had symptoms. And that got transitioned to infection versus sterile, as in nothing would ever grow. So it's the problem with the test that has led us to this medical dogma that we rely on today. But the test is faulty. And that's because bacteria are a lot like plants. They need particular nutrients in which to grow. And certain bacteria are like E. coli, which cause UTIs, are the weed of the microbial world. They will grow absolutely anywhere. They don't care really what you put them on. Other bugs are more like the fern from the lush forest. If you try and transplant that to a desert, it's simply not going to grow. It needs different nutrients, more water. It needs something else. So really, when we're talking about culture negative urines in the context of a urinary tract infection, it's really an E. coli negative urine. And my team was not the first to ask this question or to come to this realization. In fact, 10 to 40% of women who have UTI symptoms are negative by standard culture. And there was a researcher back in 1970 who questioned this. She understood bacteria, and she understood she started seeing all of these women come into her clinic that had symptoms. She was like, well, what if I just hold these samples for longer? Her name was Rosalind Mascow. In a classic women in science history story, she was ignored and ridiculed. Some critics going so far as to accuse her of falsifying the data or contaminating the samples. Now, thank you. We were very aware of the critiques that Mascow faced when she tried to publish her data. And one of the biggest critiques was that, what she sampled wasn't from the bladder, it was coming from the vagina. So, because yes, there is a huge, important microbiome in the vagina. I could give a whole other talk on that. Ask me back, I'll do it. And so the idea is when you void urine, particularly in women, the urine passes by the vagina and you pick up some of that flora on the way out. So our research team had to figure out, how do we collect urine without the risk of vulvovaginal contamination? And this is a cringy slide, I apologize. But we took voided urine, we compared it to catheterized urine, and something called a suprapubic aspirate, and that's a needle directly in through the abdomen. Yes. These women were both consented and anesthetized, undergoing surgery for a non-urologic procedure. But it was a very important study because what that showed us was that there was bacterial DNA detectable in bladders that had nothing to do with the vagina at all. And the second thing we learned was that we didn't have to poke women with needles because aspirated urine was very similar to catheterized urine. So you can use catheterized urine to look at what's going on in the bladder. But voided urine was distinct and is actually very close to the vaginal microbiome. So we published this study in 2012, and then almost immediately, we got the critique that, yeah, bacterial DNA, that's nice. That doesn't mean these bacteria are alive. It's true. With sequencing, you can sequence dead DNA. That's totally fine. So we had to come up with a way to show that this DNA represented live organisms. And to do that, we went back to our culturing protocol. And standard urine culture is actually very similar to this desert ecosystem. It is highly restrictive, only a few things grow on it. So what we did is we added more nutrients, different atmospheric conditions, different amounts of water, and different amounts of time. And we came up with this idea that we clumsily called EQUIC, the Expanded Quantitative Urine Culture. And that's our mascot. A very cute little guy. And let me give you an example. And again, for those of you who don't like bacteria, this is another slightly cringe slide, but bear with me. This is a standard urine culture. It's 1 microliter of urine that's smaller than a drop of water onto a blood agar in air for 24 hours. We expand the amount that we put on the plate, we'll hold it for longer, and we add CO2 to the environment. And you can see that this urine is not sterile. Yes, ew is usually the first reaction. And I understand, I'm a microbiologist. I think this is amazing and cool, not icky. But really, before you get too grossed out, many of these organisms are actually good for you, and hardly any of them are going to make you sick. Okay? So, what do we see? What's there in your bladder? This is a phylogenetic tree, which represents the greatest diversity of organisms ever cultured from the bladder. And the way you read this is that each branch on the tree represents an organism, and you can map the distance from one branch to another. That tells you how closely or far related they are to each other. So, what do we see? Well, we see our pathogens. E. coli, Klebsiella, Pseudomonas, they cause a lot of UTIs. But notice they make up one tiny wedge of this entire tree. The rest of it is a variety of things. Some of them are what we call opportunistic pathogens, meaning that most of the time they're fine, but given the right circumstances, they can in fact cause problems. Some are benign, like Corynebacterium live on your skin, don't cause a problem at all. And some are even beneficial, like Lactobacillus and Bifidobacterium, which are healthy in both the gut and the vagina. And Lactobacillus is even related to the same organism that makes your yogurt. So, I hope by now I've convinced you that urine is not sterile. Yes? Yes. All right. But I always get the 3 very common questions next, so I'm just going to get those over with right now. The first one, can I drink my urine? And my answer to this is if you're drinking your urine, you have other problems beyond whether or not there are bacteria there. The bacteria are not going to make you sick, but the urine's not really going to help you either. Remember, it's a waste product. It's full of salts and amines, everything your body's trying to get out of your system. So if you're dying of dehydration, would you drink salt water? Maybe not. So why urine? Ew, indeed. The next one is, does that mean I can't pee on a jellyfish sting? And again, no, but not because like the bacteria is going to affect the wound or anything. It's just the wrong thing to use. To wash away the venom, you need to try vinegar. Yeah, fun fact. And finally, does this mean I can't pee in the shower, or in the pool, or in the ocean? No. These bacteria are in such low amounts, they're not going to cause a problem. The soap's going to kill them in your shower. The chlorine will kill them in the pool. And the ocean, think of all the fish that pee in there every single day. Not to mention it has its own microbiome that you are literally a tiny drop in a massive bucket. So, how does it affect your health? Why should I care about the urinary microbiome? I spent 5 years studying the correlations between health and disease in the urobiome, so I can answer a lot of questions, but really, there's one takeaway you have from this talk. It's that if you or someone you know has symptoms of a UTI, don't let anyone tell you you do not have a UTI. And UTIs, for those that don't know, are one of the most common infections in the US, and they disproportionately affect women. 50% of women will get a UTI in their lifetime, and 50% of those will get recurrent infections. And UTIs are incredibly disruptive to your daily life. Like I said before, there's the risk of death. If the bacteria go into the kidneys, cross over to the bloodstream, they can cause sepsis and death. But the regular symptoms are pain and burning with urination, frequent urge to urinate. And when I'm talking about recurrent symptoms, I'm not just saying that someone will get it 1 year and then maybe 5 years later. I'm talking about women who have these on average 6 times a year. That's 1 every 2 months on average. There are studies where women have it more frequently than that. I've talked to women who have had so many UTIs, they don't want to go out and be social. They don't want to have sex for fear of getting a UTI. It has this isolation, social, emotional, and physical component. It's incredibly disruptive. Yet the medical system kind of shrugs it off. Anytime a woman goes into the doctor, the first thing you got to do is pee in a cup and see if you have a UTI. It is that routine. Yet, 10 to 40% of women with UTI symptoms have standard culture negative. I want to come back to the statistics. I mentioned it once before. And it all comes back to that faulty test. What does culture negative actually mean? Now, there are two types of uropathogens. There are the known uropathogens, and there are the emerging or uncommon uropathogens. And of course, the known ones are the ones that can grow on standard culture. And most doctors will recognize that list. The emerging uropathogens are very weird bugs that most physicians will be like, I've never heard of that before in my life. So where did we come up with this list? Well, from case studies, meaning that somewhere at some point there was a person close to death and a physician dug a little deeper and found this weird organism that happened to be causing the problem and wrote a paper about it so that we know about it in the literature. And the thing I want to kind of emphasize here is that these emerging uropathogens are not like COVID that the humanity had never seen before and it jumped from animals to humans. These have always been there. We just never bothered to look for them. So now they're of emerging importance. So those 10 to 40% of women probably have one of these organisms that just simply isn't being looked for. And okay, so I tell you now we can look for them and we can find them, now what? I want to be clear, I'm not advocating for greater use of antibiotics. I'm actually hoping that this research will shift things in the other direction. Because in order for an antibiotic to be effective, you have to know what you're treating. And the test simply isn't telling physicians the right thing to treat. So this lack of knowledge not only is making treatments ineffective and probably making recurrence worse, but it's also probably contributing to antibiotic resistance, because a lot of those organisms get bombarded with antibiotics and they can shrug it off and move on. So what do we do? And I personally think that UTI diagnostics and the treatment of urogenital infections is coming up to an inflection point, similar to the one that cancer diagnostics hit in the early 2000s. Well, let me explain that. Back in like the early 1900s, you had cancer of the lung, right? And there was very little they could do. Maybe they could have surgery, cut something out, or finally we learned whole body radiation. There were these dramatic things that we had to do to fight cancer. And there was a push to find the cure for cancer. It wasn't until the genomic revolution that we realized there's never going to be one cure for cancer because there's hundreds of different types of cancers. So there's going to be hundreds of different types of cures. And so now, instead of cancer of the lung, you have small cell lung carcinoma of the EGFR gene, and we know how to treat that. So better diagnostics leads to better treatment. And that's where I think we are right at the cusp of for urogenital disorders. No longer are we going to be able to say, oh, you have a UTI. That's going to be the umbrella term. And then it's going to be, you have an E. coli and Aerococcus co-infection of the bladder. Much more specific and much more precise, and therefore we know how to treat it. So once we can accurately diagnose someone, we have a much better chance of treating them and treating them for good. So with that, I'd like to take any questions.
Fantastic. My inner microbiologist is like, I have so many questions.
Yes.
And my inner feminist is so appreciative.
Yes.
All right,
and
but
yes, thank you for the previous talk. You set me up perfectly. It was amazing. Thank you.
Women's health is human health.
Exactly.
Okay. Questions. All right. A very eager person.
Hi. I was curious how you kind of make sense of what seems like a diverse array of species of bacteria causing like very similar symptoms?
The question was, how do you make sense of a diverse array of species that have similar symptoms? Fantastic question. And part of the problem is that many of these organisms haven't been studied. So E. coli has been studied for decades, but no one knows who the heck Aerococcus is, right? And so a lot of that is we need to actually learn about the pathogenesis of these bugs. But I can tell you from experience trying to get a project up and running in that exact world, funding is not there, and the willingness to support you is unfortunately lacking. So we need more funding to look at these things. Yeah, not a very satisfying answer, I'm sorry. Yeah.
Yes, you.
Thank you so much for the talk. I was just curious if you could clarify, like where the state of technology and adoption is with, like, if there are better tests. Does that mean researchers know that such a thing exists, or does that mean that like doctors are actually using them? And if it doesn't mean the second thing, like how long does it take before like for something as routine and common, but also not as serious as something like cancer,
Yeah.
that actually be adopted in practice?
Wonderful question about how are these tests going to be adopted, or if they're already adopted widely in the medical community. And the answer is unfortunately no. They are still in the research realm. However, there are a couple companies trying to push that forward outside of the academic silos, right? So and then it's the concept of when does it get accepted by physicians, right? And I, again, can talk from experience, talking specifically to urologists. It's very challenging to get traction in that field, to tell them that a dogma that they have been relying on for almost a century is false. I understand that. That's hard to accept. However, other specialties, specifically urogynecologists, are very receptive to this. And I think it has a lot to do with they're just not as entrenched in their dogmas and they're willing to explore a little bit more. So it kind of depends on the physician you're going to. If you're interested in finding a doctor that understands this science and that can do the extra work to get the testing needed, check out, actually I'm going to go to my slide, liveUTIfree.com is, like I said, a patient advocacy group. They have a list of physicians that they've been collecting, not just in the US, but all over the world for those that need them. So it is a great resource. Yes, lots of information there on, and also resources to bring to your doctor if they are at all skeptical. Yeah, in the green shirt here.
So you mentioned people with short urethras, but not the longer ones.
It's very true.
And we recently heard about Kaiser and their care for people with their genitals in the last talk. Right now, a woman complaining of UTI symptoms gets a script right away for antibiotics, no test. The male care requires a culture. Do you have any idea how many non-positive cultures are associated with UTI symptoms in males, biomales, and what accounts for the difference in care?
I can speak more to the difference in the microbiome of the two sites. I mainly focused on women in my studies. There was a few studies we did on men. Yes, you're right, the urethras in men are a lot longer, so actually it's a completely different microbiome in men. However, there is some very interesting work being done, particularly around BPH, benign prostatic hyperplasia. Don't ask me to repeat that. Which cause a lot of the same symptoms as urge incontinence in women. And we do see some correlations there. As far as getting treatment goes, it is just, even though men do get UTIs, it is a far less frequent disease in men. So I don't have, maybe you know. Do you know?
I don't.
Okay.
I wish I knew.
So I actually don't know the rates of culture negative in men. Yeah. Oh, yeah.
So it really comes down to advocating for your own health.
Yes.
And whether they tell you you're negative and you have any symptoms, it really comes down to you pushing back and advocating for your whether it's the other thing.
Yeah, I think it maps, that was a very good point. I think it maps very well to the previous talk about, you know your body, you understand your body, you need to advocate for your body in the best way that you can, and finding the resources to do that and to support you. And I'm going to give one plug before I run out of time. Quick little note here. One of the reasons that a lot of this stuff is underfunded, understudied, is because women weren't actually required to be in clinical trials until 1993. Yeah, 30 years ago. June 10th of this year is the 30th anniversary of the law that Congress passed requiring women and minorities to be in clinical trials. So my company, Evvy, is celebrating this day with Equal Research Day, and we'd like all of you to help spread the word and spread the awareness and try and help bridge the gender health gap by advocating for research, equal research in all genders. So, plug for that.
Yeah, this is something that I've run into as well in the research that I've done. It's just like, well, we only have information in male rats. I'm like,
Yeah.
why would you do that?
Because those pesky hormones get in the way of the data, obviously.
Yes, but those pesky male rats want to kill each other.
Yes.
So,
Male rats, male cell lines, male,
Yeah.
women are not small men, period. Come on,
Yeah.
let's get past this.
This is something I don't think a lot of people understand. It's like prior to 1993, it's like basically all of the medical research is wealthy white men, and that's kind of it. So, yeah. Which it's as a white man, it's great for me. I recognize that, but it kind of sucks for like the rest of the human race, and also means that the things that I discover are just not applicable to people. So, but anyway, we have another question over here. Last question.
So I'm curious with especially with these recurring UTIs, how much of that is can be prevented with like better preventative measures versus better treatment?
Repeat the question.
Yeah. The question was how many recurrent UTIs can be prevented with preventative treatment rather than recurring treatment, right?
The context for my question is I used to get UTIs 2 to 3 times a year.
I'm so sorry.
I don't know why I never learned this for like women's health or anything, but I didn't know that you have to pee after sex every single time.
There's all sorts of things that you can do in your lifestyle to decrease risk of UTIs from peeing after sex, wiping front to back. Women hear this over and over and over again. And all of that does play a role, but there is actually a lot of biology coming out that if you get one UTI, that actually primes your body in very specific ways to get recurrent UTIs. So I know, right? So you have the risk of biofilms forming in the bladder. E. coli specifically can hide within the epithelium and then reemerge. And also they're finding that the infection, the act of getting an infection changes the vaginal, I'm so used to talking about the vagina, I'm sorry, the bladder epithelium in a way that makes it more reactive and more likely to respond to another infection. So not only is it on the microbe level, but on the host level as well, there are actual changes that happen. So yes, you can do all of that as best you can and yet still have plenty of recurrence, and there isn't quite the satisfying solution other than antibiotics at the moment. I'm so sorry.
All right, another round of applause.
Thank you so much.
All right, my friends. And I feel bad about saying like, oh, my friends, because I think of you all as my friends, but also